welcome to the webinar, this webinar, where we're going to hear from 3 early career researchers from the Pacific project, part of the Freshwater Quality Research Programme. And you might have joined before, but as always, it's good to give a little bit of a background and introduction to the programme. So here on this slide, you can see that this research programme is funded via NERC and with support from DEFRA. And it outlines the objectives of the programme, which is really to increase our understanding and knowledge about the sources and behaviours of pollutants within our river systems and how those pollutants have an impact on freshwater ecosystems and how we can inform improvements to the ecological status of our UK rivers and look at ways that we can adapt and mitigate the risks and improve our freshwater ecosystem services. So that's the objective. And as part of this programme, there were 5 research projects funded. And then there's an over programme champion team, which Joe, myself and colleagues at Water at Leeds oversee. And we run this monthly webinar. And since January, we've been hearing from each of the funded projects. And this just gives you a little outline of those 5 projects. And today we're going to hear, as I said, from Pacific, which is pathways of chemicals into freshwaters and their ecological impacts, and particularly focuses on microbes. So the programme is a 4-year programme. It's halfway through. So there's plenty of ways that you can engage with us and follow progress. And this slide gives you a few of those. You can email us at freshwater@leeds.ac.uk. We have a web page where you can catch up with webinars from the series, and you can also follow us on LinkedIn and X as highlighted here. So as I said, today we're hearing from Pacific, but our next and final update this year from the projects that have been funded will be on the 19th of June when we'll hear from the long-term large-scale Freshwater Ecology Programme, 12 to 1. And so, without further ado, I'm going to hand over to our speakers today and hear how Pacific is progressing. So thank you.
Okay, thanks very much, Pippa. Hi everyone, I'm Sam MacAulay, and I'm presenting some of the work today on this mesocosm experiment as part of the Pacific project, which I ran while as a postdoc with Michelle Jackson at the University of Oxford, although I'm now based at Imperial College London. And together with me, we have Holly from the UK Centre for Ecology and Hyrdology, and Kishore from the University of Bath, who will be sharing some of the results from this project. So the Pacific project is led, as you've just heard from Pippa, by Daniel Reid at UKCEH and has project partners at the University of Bath, University of Oxford, and the Environment Agency. And the project consists of 3 work packages, which all range in scales from highly controlled laboratory studies through to less highly controlled, but more environmentally relevant field studies, surveys, sampling of water chemistry and microbes in the field. And today we're going to be presenting from work package 3, which is a balance of these 2 scales. So the advantages of a mesocosm scale experiment is that it is able to provide a controlled environment for testing effects such as those of water quality, wastewater in this instance, and other stresses as well. But it's also a highly realistic system. So it receives water from the environment and has all the physical environmental variables affecting the mesocosms and the ecosystems within them. So being able to investigate the effects of multiple stresses is important because we know that freshwater ecosystems are subject to many global challenges. And just as many of the other projects in the Freshwater Quality Programme are investigating, this project was looking at climate warming as one effect and wastewater pollution as the other, and how these 2 global challenges might interact to affect stream ecosystem, microbial diversity, and functioning. So the system that we used was this experimental system set up on 3.5-meter high mobile scaffold towers, where water was pumped from a source either upstream or downstream of a wastewater discharge, effluent discharge point. And that was how we manipulated the water chemistry. And so the downstream source was 4 times the width of the stream downstream of that discharge point to ensure that there was full mixing of the wastewater effluent in the stream. And then in order to achieve the heating, we pumped this water into a sediment trap tank to try and filter out some of the sediment, and then gravity fed it through a system that involved an electric boiler, makeshift boiler tank that then fed 40-degree or thereabouts water into some header tanks to achieve temperature treatment 2 to 3 degrees Celsius above the ambient stream temperature. And so this is to sort of simulate realistic warming scenarios by the year 2100, given that many climate models are now showing it's likely we will exceed the 2-degree warming target set at the Paris Agreement. So this setup was distributed across 4 wastewater treatment plants in Somerset and Bath and Northeast Somerset, with the idea that we wanted to capture some of the context dependency across locations. So we were able to achieve a nutrient gradient by taking 4 different locations, and also there were differences in the type of wastewater treatment. So here is a photo of the sort of mobile scaffold tower with the series of tanks feeding then down to our mesocosms on the tray, sort of on a bench at 1 meter above the ground. And as you can see, there are different types of treatment. So we had an oxidation ditch at 2 of the sites, which is visible in the background of this photo here, and also trickling filter at 2 of the sites. And you can see the mesocosms, which are sort of 2.5 liters, so quite small by a mesocosm scale, but when looking at microbial responses, they represent a very large habitat. The distributed experimental design lends itself to a spatially nested hierarchical structure where we have our 4 sites, and then within each of those sites, we have our 2 water sources, upstream and downstream of the effluent discharge point, and then we have our warming treatment with 4 replicates of each of those treatment combinations at each of the 4 sites, giving 16 mesocosms at each site and 64 replicates overall. Here, you might be able to hear some of the water flowing through the mesocosms in this video, and also see some of the other organisms that will colonize these systems. So you can see some gammarids swimming around, one there carrying another of the gammarids around. And within these systems, we added some organic matter to look at the processes of degradation. So some leaf packs that you can see attached to each here, and also some cotton strips. But in terms of responses we're looking at today, we're just looking at these microbial responses, which are sampled by taking sediment samples on 4 dates, and also sampling biofilms grown on 1 by 1 centimeter tiles within the mesocosms. Here we can see the achieved warming treatment. And this is a slide I'm very proud of, because it's very hard to manipulate water temperature in streams where you're having to capture the diurnal fluctuation. But we managed over 4 weeks at each of these 4 sites to achieve a 2 to 3 degree treatment above ambient. So that's where you can see sort of the green and the purple lines above the ambient red and blue. And also note there's a slight difference between those 2 lines as well, because the effluent discharge adds a very, very minor heating treatment itself of about 0.1 to 0.2 degrees Celsius. So the downstream source is just ever so slightly warmer than the upstream source within the heating itself. And just an overview of the samples that were collected. So as I mentioned, we collected 4 composites of 4 sediment samples from each of the 64 mesocosms on 4 dates, giving 256 samples from mesocosms, which we'll be showing results of today. And we did also collect some in-stream samples, and we collected biofilm samples on 2 dates, giving a total of 432 microbial samples. So shortly, Holly will present some of the amplicon sequencing, the 16S results, but we will also plan to do some qPCR to target functional genes and also antimicrobial resistant genes, and do a subset of metagenomics on some of the samples. And following Holly, Kishore will present some of the organic chemistry analysis that was done with targeted mass spectrometry on 32 water samples taken in the morning and afternoon on 2 dates in the experiment. So with that, I will hand over to Holly.
Thank you, Sam. Yes, so my name is Holly Tipper. I'm a molecular microbiologist at UK Centre for Ecology and Hydrology, and I've been working on the microbial analysis of the Pacific Extreme experiment. And before we get into looking at some of the results, I want to cover firstly why we're interested in freshwater microorganisms and why we want to ask these questions about these different treatment effects. Okay, so microbes are diverse and abundant in freshwater environments. You might imagine that all of the surfaces within a river might be covered in biofilms, and these biofilms can contain lots of different microbes, including algae, bacteria, and fungi. And on average, in a single biofilm sample, we'll be expecting hundreds of species of bacteria, fungi, and other microbial eukaryotes. So they're really widespread, diverse, and abundant, and a really important part of the ecology of freshwater environments. But not only that, they also have key functional roles, whether this be in nutrient cycling or biodegradation. And you can see some of those indicated on this figure here. So they're very diverse, very abundant, and very functionally important for freshwater health. How can we analyse these microbial communities and any effects we might be expecting to see on them? Well, firstly, we start with collecting the samples, and these can be in freshwater environments, can be sediments, water samples, or biofilm samples, and then we can extract the DNA from them and go through these different molecular methods to analyse that DNA. Many of these methods are sequencing based, and that can be, for example, using amplicon sequencing methods, which target a region or an amplicon, and then amplify that and sequence that to get information on the particular amplicons. And this can be very representative of taxonomic responses. And you can also use metagenomic sequencing, which chops up lots of DNA and sequences it at random, which then gives you an overall look at what's been happening in communities. Something else we can do as well is look at quantitative PCR or qPCR, and this can target specific genes of interest. And I'm not going to talk about qPCR today, but it is something else we can do to look at these. So today we're going to talk mostly about the amplicon sequencing data of a specific amplicon that we've looked at, the 16S rRNA amplicon, and this will give us information on bacterial taxa and the species composition and diversity of those bacterial communities in these mesocosms. I can't get it to move on. Okay. So firstly, before we dive into the results of the treatment, I'd like to firstly cover the sort of effects external to the treatments themselves, and that's really now looking at the site-dependent effects. So what we have in this figure here is an ordination of the beta diversity of river and mesocosm sediment communities. And beta diversity is basically just the composition of the microbial communities and how different each sample is from another compositionally. So each of these points on the figure represents a sample, and they're either coloured by what site they're based at, and also the shape represents whether it was a river or a mesocosm sample. Now, for the rest of the talk, I'm only going to talk about the mesocosm samples, but just for the example here, you can see that it didn't matter whether it was a river sediment or a mesocosm sediment. Actually, we see groupings by site. So we see that freshwater microbial communities differ at different locations. And this was the case for some where they overlap, like you can see, site B and D actually overlap quite a lot, but they are very different from A and C. Now, moving on to another thing that is sort of external to the treatments themselves, we, as Sam said earlier, took mesocosm sediments and biofilms to look at the microbial communities in the different sample types. And you can see that mesocosm, biofilms and sediments also look different. These are just from the mesocosms themselves now. So what this shows is really that there's context dependent. Sorry about that. There are context dependent effects happening that are outside of the treatment effects themselves. Now, moving on, I think I'm just going to focus now on one site. This is site B, and I'm just going to show you some of our sediment mesocosm data, because we have data from rivers, sediments, biofilms, all the different sites, all the different time points. So I'm just going to focus in on this site and look at mesocosm sediments with you. So firstly, we can see that wastewater affected mesocosm microbial communities overall. So we've got another one of these beta diversity plots now here, this ordination on the left, and we have coloured and grouped the samples by what water source the mesocosm was receiving. And you can see that the difference between downstream and upstream is quite significant. We have a divergence of those different communities from those differently treated mesocosms. And that was reflected when we look at the relative abundance of different species, different taxa. Sorry, this is phyla and class here. Different taxa in these microbial communities. You can see the difference here between upstream and downstream in both ambient and heated treatments, where we have increases in some and decreases in others. For example, increases in actinobacteria and decreases in alpha proteobacteria. So overall, we can see that at this site, we have wastewater affecting mesocosm microbial communities. And I'll just say this is a representative site, but we saw this across all of the other sites, independent of where it was and when it was sampled. So now I'd just like to show another thing we need to consider is that these sites were sampled for the mesocosm sediments 4 times, so every week. And that is shown here in these plots on the right. And you can see it's exactly the same data as is shown on the left. It's just pulled out by the date it was sampled. And what we can actually see is that although wastewater is overall having a large effects on these mesocosm communities, that was different at different time points. So you can see on the 12th of July sampling that it's not really diverging as we saw with the other sample time points, and we're not seeing that same effect. And we might think, okay, well, maybe this was because it was the first sampling round, but that's not the case, because at different sites, we've seen different days doing different things. At one of the other sites, we had the 2nd of August sampling date looking very different, and the other 3 conforming to this sort of effect. So this just really highlights how complex these kind of experiments are, and how context-dependent microbial communities can be. Now, if we look at the effect that we found from heating, we actually found that overall heating rarely affected mesocosm microbial communities. And this is just now showing the case study site B, mesocosm sediments again. And you can see that plot on the top on the left. That's the one that I recently showed you, the ordination where we group by the water source. And the one on the bottom, that is exactly the same data, same ordination. Now we're just showing it grouped by whether the mesocosm had ambient or heated water. And you can see that we can't really tease apart those different communities in this way. And we found that with other sites as well, across the board, we rarely saw heating affecting mesocosm microbial communities in terms of beta diversity. And that was supported by statistical analysis too. However, I do want to shine light on one effect that we did see from heating, and that was in some alpha diversity metrics that we saw at one of the sites. So you can see here, this is showing sediment mesocosm communities. This is the sediment richness, so it's basically a metric of how many species are present in that sample. And you can see that the difference in the upstream samples in site A between ambient and heated treatments, we have a significant increase in sediment richness in that sample. But you can see that as soon as we include wastewater effects into the mix and we look at the downstream samples, that difference between ambient and heated is then lost. So This is an example of where we did see an impact from heating, but this was one of the only types of examples. And it wasn't repeated across other sites as well. And you can see actually for alpha diversity, wastewater and water source didn't actually have much of an impact on alpha diversity either, but we are seeing those compositional shifts, which is represented in the beta diversity responses. So the key findings of the microbiology data so far are that the nature and ecology of microbial communities is complex and context dependent. You know, we're seeing those site and date effects. Yet we can see the major drivers of change come through. So those impacts of wastewater were widespread across all the sites and statistically significantly impacting. However, the functional implications of what this means, these community shifts mean on freshwater and community health aren't well understood, and that's something we'll be looking into with more analysis on function as we go forward. And I'd just like to finish with saying that, yes, we saw these impacts from wastewater, but the mesocosms were fed with water relatively near to the source, to the effluent. And we might expect that in a river, as you go further downstream, you're probably going to see those effects reduce. And we've seen similar things happen in other studies that we've done. This one is an example of an antimicrobial resistance study that we did at UKCH, also led by Dan Reed, where we looked at different distances from wastewater treatment effluent and measured the normalized abundance and richness of resistance genes. And you can see that the further away you go, that drops off. So we might expect to see similar effects happening in rivers the further away we get from the effluent outfall. So the next steps for this analysis are to assess the effects on the microbial eukaryotic communities. And we've also done amplicon sequencing for 18S and ITS. So this will tell us information about other eukaryotes, including algae and aquatic fungi. We're going to investigate effects on functional genes involved in responses to chemical exposure and stress. And this will include antimicrobial resistance, which is just another function of microbes. And we're going to do this using quantitative PCR, so qPCR-based approaches. And we're also going to look at function using metagenomic sequencing. So a subset of these samples will be sequenced metagenomically, and that will give us insight into what's happening at a community level in functional responses, not just looking at specific targeted genes like qPCR can do. And then finally, we want to combine this microbial response data with nutrient and organic chemistry data, which we have, to learn more about the chemical drivers and change and really pull that story together and see what effects we can pull out from that. And with that, I'm going to finish talking about the microbial analysis now and pass on to Kishore, who's going to talk you through the chemistry data.
Hello everyone, I'm Kishore. I'm a postdoc associate at the University of Bath. I'm working in Center for Water-Based Early Warning Systems and Department of Chemistry at University of Bath. So once we have the samples in our data, so we started with sample preparation. So it involves removing the matrix effects, so by targeting the concentrated targeting and concentrating the compounds of interest, and we do this with solid-phase extraction, and this is with the SLB cartridges, which separate polar and non-polar compounds and removing the matrix effects by the water. And then these elutes were transferred to HPLC, we have targeted mass spectrometry and then we go for targeted methods to analyze these for the compounds. So we have this liquid chromatography mass spectrometry, where we have a targeted approach of finding antimicrobial compounds for these samples, and these are all done with the 18 columns, and so we have a list of almost 127 antimicrobials and metabolites. These are the wide range of compounds which we analyzed for these compounds. And you can see the rate of antibiotics and antifungals involved in this compound list. And this is the concentration of the compounds which we studied. And each panel represents individual compounds and grouped across different sites, like the site A for orange and B for yellow and B for purple and D for green. And you can see in the top right corner, mentioning in some of the compounds NS and stars. So this represents like the significant about the concentration. It means for example, if you took on the claritomycin, like the second from the top, so it has like 2 stars. This means the concentration of claritomycin is higher in site A, and it is significantly different to the other site concentration. So in a way, among the 29 detected compounds on all the sites, some of the compounds like claritomycin, clindamycin, erythromycin, these are showing some significant difference, showcasing the concentration profiles of these chemicals, and showcasing site A and site B are having a higher concentration, irrespective of downstream, upstream, or the aim or temperature effect. And to keep into the understanding about how these varies across different factors. So we have this heat map, and I know this is a very busy map. I'll try to go through it slowly. In the x-axis, you can see different compounds, and in y-axis and the left, you can see how the different factors are involved. For example, in the left, you can see the AM and PM. This means it's representing the morning and evening of the samples, and then the warming, this is telling us if temperature is considered, and then there is this wastewater source, the pink is for upstream, and green is for downstream samples, and then the last is WWP. That means sites, for 4 different sites, and this is in the left, and you can see there is 2 clusters, one in the left and then one in the top. So the row-wise cluster helps us to understand how these chemical profiles clusters the compounds. For example, in the top, you can see the site C, site A, which is having definitely significantly independent profile. So this means so it has significant chemical profile which differentiates with other 3 sites, and so this is what we were able to find, and so this also helps us to understand in the cluster from the top, we can see different chemicals having a significant profile. For example, in some of the compounds like claritomycin or fluconazole having higher concentration across these all sites, and so this gives us an understanding how these effects are contributing towards the chemical profile, so that we want to understand if these chemicals profile is significant towards site, and if there is any significant towards the wastewater treatment site, and then the source of the water, and but from this the heat map is still vague, so we have to see the significant difference between the site from the top row, it differentiates the site A is having a different profile, but not gives us some more information. So for that, we don't throw the PCA plot. So in the PCA plot, you can see the in the right hand side, so you can see the 4 different sites, and you can see the site A is completely diverse towards other 3 sites, and the other 3 sites, and the other 3 sites, site B, C, and D, having some overlap. So, and this could be one of the reasons, about the size of the catchment. So you can see in the left hand side the map of each catchment map, and this is wastewater treatment plant catchment map, and you can see the site is significantly bigger size, and this could be one of the reasons, but still there need to be more investigation need to be done. For example, you need to understand the flow, and if there is any dilution factor involving in this, so this still need to consider. But this from these results, be able to clearly I understand the site A is having a significantly different profile, and we try to introduce to see the PCA to see if there is any effect on downstream and upstream, and also temperature. So you can see these are the same sampling points, but now they are grouped based on upstream and downstream. And when you compare an overall picture, so there is no big difference between upstream and downstream except for one point, which is giving us some difference. But when you focus only on the bottom level, which are representing the site A, so you can see there is a difference. But when you compare overall upstream and downstream, we don't see much significant difference only for a few compounds. But when you see the warming effect or like ambient and temperature, like so it's completely overlapped. So we don't see any significant difference between the samples. So these are the findings from us, and yeah, from this concentration data, like among the 29 pharmaceuticals, because these are the 29 pharmaceuticals able to higher than the limit of detection and be able to detect these from the among the 127 pharmaceuticals and metabolites in our list. And from among these, from the 29 pharmaceuticals, we are able to understand that. Yeah, we're able to understand that like site A shows like highest level and concentration, and this site A have like dominant effect like based on some of the compounds, and when compared to site B and C and D. And in terms of upstream and downstream, there are like two compounds which having like a significant amount of difference based on their P values. But yeah, it is like only partially supported, and yeah, these are like similar to what Polly have observed, like in terms of the effect of upstream and downstream. And we also don't see much significant difference because of the warming. These are the results, and talking to Sam.
Okay, thanks very much, Holly and Kishore. So just to present a final sort of overall summary of the results that we've shared today. The clearest effects that we saw across both microbial community responses and the chemical profiles were those of the site. So site-dependent responses likely driven by population size and catchment size, as Kishore was discussing earlier. We did see widespread wastewater effects, particularly on microbial communities, but this was only supported by a few specific chemicals. And overall, heating rarely had an effect on microbial communities, and there was no effect evident on water chemistry. So the findings from this experiment provide supporting evidence for other studies, which have shown overall that land use is the dominant factor when compared to climate or specifically warming effects on freshwater ecosystems, which is a more and more supported result from meta-analyses of multiple stressor studies and sampling of field surveys, where we see that really land use is driving more of the variation in ecological and chemical responses when compared to climate effects. So with that, I think we can open up to questions and discussion, and I believe I'm handing over to Joe.